Hey everyone, welcome back to Cell Culture 101. Today we're going to dive into a really common problem in cell culture, poor cell growth. If you've been growing cells for a while, you've probably encountered this at some point. It can be super frustrating, but don't worry. We'll go through some of the most frequent causes and the solutions to get your cells back on track. Let's start with the first issue, poor quality stock culture. This one is a big culprit behind poor cell growth. If your stock culture wasn't frozen, stored, or thawed correctly, it can lead to a lot of issues down the line. For example, when freezing your cells, you need to ensure the freezing process is slow, about 1 to 3 degrees Celsius per minute. If it's too fast, ice crystals can form and damage your cells. And remember, stocks need to be stored at temperatures below minus 80 degrees Celsius, ideally in liquid nitrogen, to keep them viable. When thawing your cells, it's crucial to do so rapidly and immediately remove the cryoprotectant to preserve cell viability. Now, let's address slow cell growth. This one is tricky because there are several factors at play. One of the first things to check is whether your cells have been passaged too many times. If you've been using the same stock for a while, it might be time to refresh it. Cells that have been passaged too many times can lose their growth potential. Ideally, you use a new stock that's been subcultured fewer times to maintain healthy growth. Another thing to watch out for is cell confluence. If you're harvesting cells that are already too confluent, they'll have slower growth. Always harvest your cells before they reach 100% confluence. Also, don't forget about your media. Poor quality media, serum, or buffers can seriously hinder cell growth. If your reagents are outdated or improperly formulated, your cells will feel the difference. And remember, carbon dioxide levels play a huge role too. If your media contains bicarbonate, the carbon dioxide concentration needs to be correctly adjusted to maintain pH levels. Without this balance, your cells won't grow properly. Finally, let's look at uneven cell growth. This is a problem that can happen when the cells don't grow uniformly within the vessel. The most common causes are inadequate mixing, uneven temperature distribution in the incubator, or the presence of air bubbles. To solve this, start by making sure your cell and media mix is even. A gentle vortex or pipetting can help ensure uniform distribution. If you're seeing uneven growth within a single vessel, it's usually because of poor mixing. Another issue could be temperature variation within the incubator. If your incubator shelves aren't level or there are hot spots, it can cause cells to grow unevenly in the same or different dishes. Make sure your incubator is level and try to avoid stacking your vessels too high. And of course, keep an eye on air bubbles in your culture medium. If there are bubbles, you might notice areas where cells aren't growing properly. To wrap it up, poor cell growth can stem from several different issues, but once you identify the root cause, it's usually pretty straightforward to fix. Whether it's optimizing your freezing and thawing protocol, improving cell attachment, or adjusting your growth conditions, these small changes can make a huge difference. Thanks for watching, and I hope these tips help you troubleshoot your cell culture problems. If you found this video helpful, don't forget to like and subscribe. See you next time.